All right, so now that we've went through our introduction to cellular division, we're now going to go into the cell cycle and the mitosis process itself. And so before we get into that, we need to first really define what what exactly is happening in mitosis. And mitosis is a division really of the DNA and the chromosomes. And so we're going to define chromosomes. We're going to talk about what they are. We're going to talk about their structure, and then we're going to talk about how they get divided in mitosis itself. So in eukaryotic organisms, so eukaryotic chromosomes, the chromosomes are made up of what we call chromatin. So this chromatin, these are consisting of fibers that have roughly equal amounts of DNA and protein molecules, and we'll show a figure of that in the next couple of slides. And these protein molecules are really important because they help organize the chromatin, and they help to control the activity of the genes themselves that are located on those chromatin fibers. And so a lot of the times the chromosomes are going to be these really thin fibers that are just floating around in the nucleus, and they're not really going to be condensed into the structures that they are like they are in mitosis, where you can actually see them with a light microscope, which is this instance here. So these are plant cells that are just before the division process, and these plant cells have been stained so that you can see the condensed chromosomes. And so this is a nucleus on this cell that doesn't have those chromosomes condensed. Okay, so we mentioned that these chromosomes will have their material that interact and are wrapped around proteins. And the proteins that the chromosomes wrap around are what are known as histones. And these help to package that DNA to basically keep it tight and compact inside the nucleus when it exists as that chromatid. And you can have groupings of these proteins that are known as nucleosomes. So here we have your DNA, the double helix, okay? And you have your histones, which are these purple balls, and the DNA will wrap around these histones, these proteins. And you can have groups of these packages, and each group will be known as a nucleosome. So they have different, several nucleosomes that are running through this this chromosome strand. And if you look at it under a microscope, it actually looks like beads on a string, and so that's kind of what they refer to it as, is beads on a string. And so you have your DNA that's wrapped around these histone proteins that is then kind of compressed into this nucleosome, and then you can package these nucleosomes really tight together, and you end up with a chromatid. And in this case, we have a sister chromatid. This is a chromosome that's been duplicated, and so this side is identical to this side. And those two sister chromatids are held together at this, what's called the waist, by the centromere. Okay. And before the cell division process happens, before mitosis happens, the DNA is copied. So each individual chromosome is copied, is duplicated, and you end up with those sister chromatids that you see in this figure here. And so, like I said, they have identical genes on those sister chromatids. And again, those sister chromatids are joined at the waist, which is called the centromere. So that's where those sister chromatids are joined together. All right, and so when cell division happens, the sister chromatids will separate from one another. So during mitosis, those or meiosis, so it would be meiosis two, which you'll learn about. So in mitosis or meiosis two, the sister chromatids will separate from one another. And once it's separated from its sister, so its pair, each chromatid is basically considered a full chromosome and is identical to the original chromosome. Okay, and so this figure here will hopefully help that make more sense. Here we have a chromosome. It becomes duplicated. So now you have two identical copies of that chromosome, and this is your sister chromatids. And the sister chromatids are joined at the waist here. Okay, so that's the centromere. And once they go through the process of cell division, again, it can be mitosis or it could be meiosis two, the sister chromatids are separated from one another. And then they end up being individual chromosomes that are genetically identical to that original parent chromosome. Okay. And so now that we've pretty much learned about the DNA and how it's structured in the nucleus, how it um, condenses down into your chromatids and then your sister chromatids, we're now going to talk about the actual phase in the cell cycle and the process that the cell goes through during its life. And so the cell cycle is basically these series of events that occur once a cell is formed. And so basically from the time of birth to the time it divides and creates its own daughter cells. And so the cell cycle is basically this lifetime sequence. Okay. Most of the cell cycle is spent in what's called the interphase. And so this is 90% or so of the cell cycle. And so interphase is made up of three separate phases, and we'll talk about those in a little bit. Basically, during interphase, the cell performs its normal functions, so its normal cellular processes, so cellular respiration. If it's a plant cell, it undergoes photosynthesis, etc. It also doubles everything. So it doubles its organelles, it increases in size to hold all the new organelles, and it also duplicates its chromosomes. So it basically doubles everything that exists in the cytoplasm. So here we have an image of the eukaryotic cell cycle. The light blue here is interphase. And interphase is separated into three different phases. You have the G1 phase. G stands for gap, so the first gap phase. The S phase, which is the synthesis phase. This is where your DNA synthesis happens, so your chromosomes are duplicated. And then the G2 phase, which is the second gap phase. And then after interphase, you have your division phase. And in terms of mitosis, this is the mitotic phase. If it were gametes, it would undergo meiosis. And so with these phases here, your cells, remember, are really active during interphase and during those three phases. Okay. And so even though the G phases are called gaps, those cells are still active, they're still growing, and there's a lot going on within those two G phases. And if you pay attention to this figure in the first gap phase, right, there's one copy of that chromosome. Okay, so there's hasn't been duplicated yet. And then once you pass the S phase, that synthesis phase, now each chromosome is duplicated, and they're now sister chromatids. And they stay sister chromatids up until that division phase. And once that division phase happens, then you end up with your full-fledged individual chromosomes, that are genetically identical to the original parent chromosome. Okay. All right, so once you leave the interphase portion of the cell cycle, the cell undergoes the mitotic phase. And the mitotic phase has two processes that kind of overlap one another. The first is mitosis, which is where the nuclear contents divide evenly into your two daughter nuclei. Okay, so this is basically dividing the chromosomes. Cytokinesis is where the cytoplasm and all of the organelles will split into the two daughter cells. So this is where they're separated, that material separated into the two individual daughter cells. So the overlap or the combination of mitosis and cytokinesis is what produces two genetically identical daughter cells from a single parent cell. And so here's a figure looking at the process of mitosis. And there's kind of a lot going on here, and there'll be kind of a lot of stuff that you'll need to remember throughout this process. And so we're going to kind of work through this. So first, we have interphase. Okay, so interphase is right before your cellular or the division process happens. So you have uncondensed chromosomes, you have centromeres. Remember, centromeres are part of the cytoskeleton organelles. Those are the microtubules, microfibers, those sorts of organelles that help keep structure and move organelles across other organelles across the cell body. You have your nuclear envelope, then you have your plasma membrane. Okay, so these are kind of the more important structures for the mitotic division, and we'll also talk about these in meiosis. After the cell enters the division phase, it will enter prophase. So pro being before, and phase, the first phase, or before all the phases. And so prophase is the first phase of the mitotic process. All right, so prophase, the first phase in mitosis. In prophase, we have a couple of different things going on. The first important thing is that the nuclear envelope is breaking down, and it's basically going to kind of disintegrate and go away. And this is important because you can't separate your sister chromatids unless that nucleus is out of the way. And so that nuclear envelope breaks down. And then you have your mitotic spindles also forming out of the centromeres. Okay, so those tubule-like structures. And these are going to grab onto those sister chromatids during this process of cellular division. What's also happening in prophase is that the centrosomes are migrating to the poles of the cell. And so you can see a centrosome here and a centrosome here. And that's because the spindle fibers are going to grab on and to those sister chromatids. When they pull them apart, they're going to pull them to each end of the cell, which is where each of the daughter nuclear material is going to be located. Okay, and so you have your nuclear envelope breaking down, your spindle fibers forming, your centrosomes migrating to the poles of the cell, and your sister chromatids condensing into those chromosomes like you see here. And now when we move into metaphase, those condensed sister chromatids line up along the metaphase plate. Okay, and so the metaphase plate is basically the equator of the cell. Okay, so this area here. So you'll have one sister chromatid on one side of the metaphase plate and the other sister chromatid on the other side of the metaphase plate. And so metaphase is lining up of those chromosomes, the sister chromatids, along that metaphase plate. And with the metaphase plate, it could be in any direction, right? It could be vertically, it could be horizontally, it could be at a diagonal. And so as long as it's lining up along the equator of the cell in kind of any plane is fine. Because you're just wanting those sister chromatids to be pulled to either pole of the cell once they're separated. Okay? Now, once we go from metaphase to anaphase, this is where the spindle fibers start to shorten. So they start moving away from the metaphase plate and back towards the centrosome. The shortening of these spindle fibers breaks the hold that exists at the centromere of the sister chromatids, so the waist area where they're attached, and breaks those sister chromatids apart from one another. So now you have an identical copy of the chromosome going to each end of the pole of the cell. And so in this example, you have 1, 2, 3, 4 chromosomes, each duplicated. And so whenever they get pulled to each pole of the cell, you'll have 4 chromosomes on this side and 4 chromosomes on this side that are all genetically identical. Okay? And so anaphase is the physical separation of those sister chromatids from one another and being pulled towards the pole of the cell. Okay? Then in telophase, so a lot of times you'll see telophase slash cytokinesis because these are kind of happening together. But in telophase, you have the reforming of the nuclear envelope. You have the chromosomes to uncondense. You have the spindle fibers to go away. And you also have the physical separation of the cell in cytokinesis. So cytokinesis is where the cell divides itself into 2 cells. And so you have a cleavage furrow in animal cells to form, which is this indentation here. And that leads to the physical separation of these 2 cells, which eventually will form 2 daughter cells. Okay. And so this is the process of mitotic cell division. So you have your prophase, your metaphase, your anaphase, and your telophase. And you need to know all of the key details that we just discussed in each of those phases. And like I mentioned, right, the chromosomes and their movements are based on that mitotic spindle. So those microtubules, and those are physically separating those chromosomes. The spindles are what's moving the chromosomes across the cell. Cytokinesis, also, like I mentioned, begins during telophase, and it's physically dividing the cytoplasm and the cell itself. That process of cytokinesis is different for plants and animals. So notice I said that this was an animal cell because it has that cleavage furrow. In plant cells, that happens a little differently. So in animal cells, cytokinesis is known as cleavage, and it begins with that indentation, that cleavage furrow at the indentation of the equator of the cell. And this is a little bit different compared to plant division. Okay. Before we move on, though, I want you to think about this scenario. So if an organism called a plasmodial slime mold is one huge cytoplasmic mass with many nuclei, so it has multiple nuclei, how could this arise? And so how could you get a cell with multiple nuclei? So think about that for a minute. Pause the video. All right, so basically what you would have happen is, let's say you have mitosis occurring multiple times, but you don't end up with cytokinesis. So you have multiple nuclei with identical chromosomes, but cytokinesis never happens, so you don't get a division of that cell. And so all those nuclei stay within that parent cell. And so that's how you could get a cytoplasmic mass with many nuclei. Okay, so in animal cells, we know that cytokinesis, also known as cleavage starts with that cleavage furrow. And so this is a micrograph, so a scanning microscope, where we're looking at the cell cleavage furrow here. So this is a cell starting to divide in two. So this is basically the portion of the image we're seeing here. So the top or bottom portion of the cell. And then there's microfilaments. You can almost think of like a rubber band being put around the cell and it constricting until it kind of pinches the cell in two. And so that ring of microfilaments will contract and contract until that cell is split into two daughter cells. Now this is functionally different from how a plant cell undergoes cytokinesis, because a plant cell actually uses what's called a cell plate. And that cell plate forms from a membranous disc that is a culmination of multiple vesicles kind of building together. And so the cell plate forms in the interior of the cell and actually grows outward towards the plasma membrane. And so eventually that cell plate, that membranous disc, will fuse with the plasma membrane, and it will separate the components inside the cell, and it will make two daughter cells from a parent cell. So here we have an image of this happening. So here's an actual micrograph, light microscope image of a plant cell that has a cell plate forming. And the way this works is you have your parent cell, you have the nuclear material already separated, then you have vesicles forming, membranous vesicles forming in the interior portion of the cell. These vesicles will elongate and grow until they connect, forming a cell plate. And that cell plate will keep growing outwards until it connects with the plasma membrane. And then now you have two new cells that are separated from one another. Okay, and so that's it for our mitotic process and the cell life cycle. In the next video, we'll talk about cells dividing out of control. We'll talk about cancer and what that comes from. Okay, so we'll see you in the next video.